Thank you. And salve and good morning and greetings from the coast of Maine, where it's very early morning. In any case, I want to thank the organizers, particularly Riccardo Manzotti and Stuart Hammeroff, for generously allowing me to contribute via Zoom. I apologize as well for not being with you all today. And thanks also to Anna Lucentini and Giovanni Bombazzi for facilitating all of the the video connection. So let me get started here. I'm just going to share my screen. Okay, here we go. And so, all right.
I was too much optimistic. Daniel, can you hear us? Hello? No. Hey.
Sorry. Okay, here we go.
Okay.
I apologize. All right. I think we're good, right?
Right now, yeah.
Okay, let's get going, and I will share my screen again.
Okay.
And sorry for the rough start. Here we go. Seem to be having trouble here.
Yeah.
All right.
Yeah.
Give me a second. Okay, so here we are.
Yeah, it
Let
works.
me. So you all can see this, I take it. All right. So the talk has sort of changed its character because it has started off as a concurrent, and now it's a plenary. But I want to bring evolution into this picture that I'm going to present here. So first of all, let me give you an idea of how I'm going to do this. I'm going to talk about evolutionary history and the utility of what I call analogical reasoning, that is looking to modern organisms to understand past events or past organisms which are at best fragmentary, both in terms of physical presence and any kind of relic of behavior, and the reverse as well. I'm going to turn to the Cambrian, the so-called Cambrian explosion, where I believe there was sort of a revolution that occurred in terms of sensory perception. I'm then going to move on to animal eyes, distance vision, and how that sort of changes or revolutionizes the representation of space and time in the nervous system. And then I'm going to briefly, very briefly, and this will be familiar to some of you who've seen my previous talks, I'm going to sort of offer what I believe is a sort of a reasonable definition of consciousness. And then I'm going to move to the case of the octopus. We'll talk about to some degree about physiology, functional anatomy, and behavior, which I believe we can argue support a form of sensory consciousness. And finally, I'll give you sort of a broad brush, a plausible natural history of awareness. Okay, so I'm going to start with this weird allegory, which was sort of passed on to my father, from my father to me, and it sort of illustrates a point that I would like to sort of make in a very strong way. So I'm going to tell you a little story. It's about a New Yorker. He's on his way by subway home to Brooklyn from Manhattan, and he's finding himself working himself into more than a little bit of a rage because he believes that his wife has been having an affair behind his back. He rushes home on the subway as fast as he can. He climbs the 5 stories to his top floor walk-up apartment. He's looking for what he believes to be his wife and or her lover somewhere in this apartment. He can't find them. He rushes out onto the fire escape. He sees a man below wiping his brow, and a flush of anger ensues. And in a rage, he grabs the nearest heavy object, which happens to be an old refrigerator, and drops it off the fire escape onto the head of the man below, and immediately dies of a heart attack. The scene shifts to the pearly gates of heaven, and St. Peter is looking down on three men. And so St. Peter says, well, gentlemen, you know why you're all here. We all realize that this is a new stage for you. And you're all coming in. There's no problem there. You're all entering heaven. But I need to know, first of all, how you all ended up here. So guy number one, what's your story? What happened? Well, St. Peter, I was beginning to believe that my wife was having an affair on me. I rushed home, and in a fit of rage, I saw this guy below me on the fire escape. He was wiping his brow, and I got really, really angry because I thought, wow, this must be the guy. This has got to be the guy. I picked up a refrigerator, dropped it off the fire escape on this guy, and then died of a heart attack. St. Peter said, okay, great. We've got your story straight. Guy number two, what's your story? Well, St. Peter, it was a very hot night in New York City. I went out. We have no air conditioning. I went out. I was wiping my brow. I loosened my tie. I was trying to get some fresh air outside. And all of a sudden, this refrigerator dropped on my head. St. Peter said, that's quite interesting. Well, guy number three, what's the deal with you? Well, St. Peter, I don't know. I was just sitting in this refrigerator minding my own business. Okay, well, what's the purpose of that strange story? The purpose is to sort of highlight the idea that classic evolutionary science in the classical sense, leaves you in a different place than what we think of sort of as conventional empirical science, in the sense that the experiment has already run. It's perhaps millions of years old, and we can only reconstruct what happened from previous events. We can only understand it after the experiment has unfolded and is long gone, and hence this refrigerator story. So let's talk about this important idea of analogical reasoning plus notions of convergence. And so we all understand evolutionary convergence is the idea that similar environmental conditions give rise to similar biological adaptations. I will also promote the idea that powerful, perhaps universal constraints are in play on animal form and function, and we see this consistently in the evolutionary record. Evolutionary convergence was once thought to be something that was relatively rare. We don't really believe that anymore. And it's amazing how similarities pop up across sort of a broad range of phyla that are otherwise not connected by single ancestors, but their last common ancestor was hundreds of millions of years ago, and we'll see that with the octopus. So I'm going to bring in the idea of the importance of focusing eyes. In my mind, I believe that the innovation in vision that focusing eyes afforded really changed the landscape, and also meant that a number of things were happening, neurobiologically speaking, behind those focusing eyes. And we're going to talk about that in a bit. So focusing eyes didn't appear without the requisite neural substrates for processing and reconstructing a complex visual scene. Another idea, which I think is kind of important, despite some really rather remarkable differences in the so-called phenotypes of neurons. A neuron is basically a neuron, regardless of species. Insects, you'll see tons of so-called monopolar neurons in the case of insect brains, insect nervous systems, and other invertebrates as well. There are many different types of neurons, but basically neurons are carrying out a certain function universe that's universal, effectively, right? Transmitting a signal, processing in groups or in sort of coordinated assemblies, processing somehow some aspect of the world coming in or some internal signal. All right, so finally, familiar brain circuitry. Now, we see this in the vertebrates, across all the vertebrates, of course, and we can make arguments, especially in the case of higher vertebrates, from homology. That is, that you find cortex in all mammals to greater and lesser degrees. You find hippocampi in mammals and in other higher vertebrates, other vertebrates, period. You find all these different brain structures, and they're common in all mammals. When you go beyond the vertebrates, that familiarity sort of begins to diminish. But you do pick up on some very, very basic similarities, which point to the idea that there are only so many ways that nervous systems can encode aspects of the world, and in particular space and time, as will come into play when I present this argument about the importance of vision in the origins of consciousness. So, evolutionary convergence is sort of a well-known idea. It's been around for a very, very long time. This is a classic example. You can see this in the appendicular skeleton of various higher vertebrates, including humans, birds, and a mammal like a bat. You can see that a similar function has arisen in the case of birds and bats, and also in the case of a reptile. And there's a similar function being carried out, flight. But the way you get to that function is somewhat different, but still you can pick up homologies in the skeletal structures of all these different animals all the way on up to human beings. So, of course, they're not using their arms to flap around and fly. All right. So let me set the stage by talking briefly about the Cambrian. There was an explosion of invertebrates, an explosion of all kinds of diverse life and complex life, I might add. In the case of the invertebrates, it's really rather remarkable. It's this sort of efflorescence of different body types. And you see in the examples on the left, you see some really, really strange animals, which, you know, we don't have modern counterparts for. We haven't found anything that really resembles them, but we can see certain things that are coming to the fore. You can see in a couple of these cases, you see animals that have a pair of eyes. Now, they're fully functioning eyes. They may be compound eyes, but they're still eyes. And eventually, you have focusing eyes as well. But when you're dealing with the reception of light, you're dealing with some basic attributes that eyes and the neural circuitry behind them are interpreting and perhaps deconstructing and then putting back together in some sort of representational form. So reflection, transmission, absorption, scattering are sort of the common sort of properties that you know, all forms of animal eyes have to deal with. Brighter or dimmer, there are various properties of light, spectral properties, and they may even yield differential wavelengths, which many animals can discriminate, and some animals, including us, can discriminate as colors. Polarization is another property. We're not going to really talk about it here, but animals like octopuses are capable of seeing polarized light, making some sense of that polarized light, and using it in some sort of adaptive fashion. That's not uncommon. We don't have that capacity, but birds do. Certain other vertebrates, but many invertebrates have this capacity. Cambrian ecology really comes to the fore in the argument that I'm presenting here. You have mobile animals that are descended from sessile or stationary animals that are more or less pinned to the ocean floor, and this happens probably in 10 million years or less. You have mobility of animals at photic depths. When I say photic depths, I mean depths at which light is really a player, that you can start to discriminate objects visually. And when animals got more mobile, they could exploit more far-flung food sources, and they could engage in novel predatory strategies, and there was sort of an arms race of innovation. And eventually, you see faster locomotion, armor, peptide mimicry of neuromodulatory signals, all kinds of defenses, as well as refinements in vision. All right. So the evolution of eyes it's sort of an interesting period in the Cambrian. Distance vision is something, in fact, that's totally apart. It's sort of unusual, and it's very important when you look at the history of eyes, animal eyes. And so eyes, even simple eyes, are not merely light-sensing organs. They're able, in their way, to discriminate spatial properties of the world. And this is by virtue of the fact that there are pigment cells that separate photoreceptor cells from one another physically and provide sort of boundaries. So you can sort of interpret the eye as a sort of a spatial array, and that spatial array is sending signals into a central nervous system into the brain, and those spatial properties are sort of more or less preserved in many brains, at least. So there are many different the light motifs for eyes are many and varied in the Cambrian and onward. We see modern examples of animals with quite different but very sophisticated forms of vision. There's the case of the male jumping spider. They also have a focusing eye, but the architecture is completely different than anything like, say, a camera eye that we're accustomed to in invertebrates and some invertebrates. You have mantis shrimp who have 13 to 15 different kinds of photoreceptors. Arguably, their color or their perception of different wavelengths of light is more capable of fine discrimination than even our eyes are capable of. So here's just a sort of a picture of the different sort of types of light, of eyes, and their reception of incoming signals. All right. We also have sort of a convergence of function in the central nervous systems of various animals. And I point to the hippocampus as sort of one example. So again, I said across the vertebrates, hippocampi are, they're all over. They're in all the vertebrates. In the case of a honeybee, you have a particular structure called the mushroom body, and this has been quite well and favorably compared to the hippocampus in the vertebrates. It plays a role in memory formation, perhaps in recall as well. And then finally, you have the vertical and median superior frontal lobe complex in animals like the squid and the octopus, and the so-called coleoid cephalopods, and all cephalopods, but in particular, coleoid cephalopods like squid, octopus, and cuttlefish. And these areas, this particular region, this complex is thought to, function in much the same way that, say, a mushroom body or even a vertebrate hippocampus functions in terms of memory formation. All right, so we have to be careful, because when we look at even relatively modest nervous systems, they actually can generate some very impressive behaviors. You can see that in the case of the fighting fruit flies that are fighting over food in a dish. You can see that in waggle dancing honeybees, giving their conspecifics an idea of where food source is. You see that in the case of a jumping spider throwing itself at a bee, first observing and then moving quite accurately to capture that bee. And then finally, you see this in the chevron pattern, the signaling of a cuttlefish. It's really, really quite fancy and remarkable behavior. So these are quite sophisticated, but we're talking about something a little bit different here. So how much and what sort of a brain is required for conscious experience? That's a question I ask constantly. What about animals without backbones, like the octopus? All right. So I'm going to provide a very basic definition for what I believe primary or basic sensory consciousness is. I would say we have to start with the stitching together or binding of many closely contemporaneous sensory threads into a coherent unified scene and the persistence of that scene in memory. This requires a link between perception and memory. Conscious states can be looked at, I believe, as so-called bound scenes that offer content which is correlated in time, that is closely contemporaneous in time, beyond that arising from, say, segregated sensory inputs, sort of an integrated information of sorts. I'm invoking that in a very general way, not as regards to the particular theory of IIT. And this particular view suggests that a surprising number of non-human animals are capable of subjective experience. All right. So there are certain things that I think we can look toward the example of the mammals in particular and sort of apply across the board, even beyond the vertebrate line, when we're talking about the necessary substrates for consciousness. So first of all, I think we need to look for brain regions that function like thalamus and cortex. That is, that there are areas that perform the same sorts of function in widely different animals. In the case of thalamus, pulling in signals from the world, from various sensory modalities, and perhaps communicating with cortex to cortex those signals, but then having sort of an ongoing conversation with cortex and resulting in some sort of a reentrant or recurrent kind of signaling. I think that's important. That's something that we've been observing in what we take to be conscious behavior in all kinds of mammals, including humans. Dynamic brain activity of a particular variety or a particular pattern. The idea that we can observe firing of neurons across the cortex, well, we can perhaps observe something similar across brain substrates functioning very much like cortex in a completely different sort of an animal, in an invertebrate. And that goes to the idea that I don't think that there are 9 ways to skin a cat as far as actual activity patterns go in nervous systems. I think we're probably going to find there are sort of basic repertoires of firing patterns that look quite familiar regardless of the species, if we're talking about complex animals and their behaviors. The ability to make sophisticated discriminations. Of course, many animals can make discriminations, and this may be a matter more of degree than of kind. But in any case, the ability to make the kinds of sophisticated discriminations that we often associate with consciousness suggests a very deep connection between perception and, I believe, memory. All right. So there are a number of important things that we can look at, I think, across species that are all kind of important in framing an argument for the existence of consciousness. I believe that all of these are sort of requisite for conscious experience, for a brain experiencing the world in a conscious sense. So fast sensory channels, vision is one. I would put olfaction kind of at the other end of the scale. It can be very sophisticated, particularly in certain non-human mammals, dogs, among others, but it doesn't have the speed in terms of sort of putting together a signal, acting quickly on that signal, and also the signal itself being propagated in such a way within the air to allow for the kind of sort of, I guess, rich model making that goes on when we're talking about something like distance vision. I think we need to look at a density of cross-modal or submodal sensory inputs, because that, in fact, is something that we've seen in mammals and other vertebrates that arguably some of us would say are conscious. Fast integration of discrete sensory signals, which inevitably or eventually yields what we call perceptual unity, the ability to bind together all of these sensory inputs into a unified scene. And then finally, working memory. That's quite important, I believe. And last but not least, the brain circuitry that links perception and memory. Okay, so let me do this. So let's talk about exploring consciousness in far-flung species. I've addressed what I believe answers the question, at least in a basic way, what is consciousness? We have practical questions. What are the structures and functions necessary for consciousness? How do we study it? Which animals should we study? Invertebrate consciousness represents an interesting example because there's a wide variety of invertebrates with very simple to very complex nervous systems and sensory apparatus. So many invertebrates have sophisticated faculties, including learning and memory. Integration, arguably not as much, and behaviors as well. And polioid cephalopods have these very sophisticated faculties. They have complex nervous systems and circuitry that can kind of support the above. We need to come up with some sort of tractable and systematic approach. I've taken to the idea that we should map the octopus visual system because I think it's our way in to exploring perhaps conscious experience in these animals. And we really should try to work out the neurophysiology of visual perception and memory in cephalopods. We have to be careful of the so-called pitfalls of parsimony. In terms of evolution, that doesn't necessarily work. The simplest solution isn't necessarily the solution that's sort of most often taken. It does happen, but it's not, we can't, have Occam's razor all of the time. There are constraints on network properties. Do the same general principles apply to all complex nervous systems? Well, I believe that there are, as I said before, there are constraints on properties. I think we'll start to identify what look like very, very familiar sorts of signaling patterns when we start recording in great detail from the nervous systems of animals like colloid cephalopods. All right, so let's get to the octopus. So there's structural and functional homology among humans and other vertebrates, as I said before. We can see this quite clearly in the case of a human and on the right, a zebrafinch. All of these familiar structures, they do seem to have their homologs. And more recently, of course, in the case of birds, it's been argued that effectively there is homology in terms of something like a cortex, that the same sorts of cells during embryogenesis inevitably give rise to something that's functioning very much like a cortex in a bird. So that's not up for much debate these days, I believe. But when we get to cephalopods, it's sort of a different story. We've characterized some of these regions in terms of their function, but in terms of the gross structure, a lot of it is quite unfamiliar to us, except when we get sort of finer grained or more detailed studies bring these to bear. Then we start to see some similarities which are interesting, some architectural similarities. I'll kind of bear on that a little bit later. So we've identified certain structures, certain functions of these various structures in the cephalopods. The vertical lobe and superior frontal lobe, this particular complex is again important in learning and memory across different sensory modalities, vision and certainly tactile among others. And we started to record brain activity in behaving octopuses. This is an earlier example by Graziano Fiorito and Benjamin Hockner. They really sort of innovated, and they did the first sort of good recordings from octopus brains. But it was a single electrode. It was a stainless steel wire that was inserted in a particular brain region, and after doing this in perhaps dozens of different animals, it was possible to produce better maps of function in the octopus brain, both in terms of the sensory end and in terms of the motor end of things. Now, lately, there's been a major innovation. A group led by Tamar Gutnick and Michael Kuba were able to record brain activity in untethered behaving octopuses. In that previous example, you understand that it was a wire approach, and eventually the octopus finds the wire and often pulls that wire out along with it, a part of their nervous system. So this avoids that. It's planted on board. It's a data logger. So it's effectively wireless. You're not communicating wirelessly via, via Bluetooth or anything like that. But you have a data logger on board, and then you can sort of download the information. So Gutnick and her colleagues managed to actually do this in the case of some octopuses, and they started recording brain activity. And what they found, interestingly enough, and this is more of a proof of principle study than anything else, but they did start to find patterning, sort of a pattern of firing during certain sequences, certain events that resembled the vertebrate case. And among other things, in the case of animals in resting states, we can start to pick out very familiar leitmotifs that we've seen before in the vertebrate case in a dramatically different invertebrate like the octopus. And this is just beginning. This is just a very early salvo into what I believe is going to be a very promising direction in research. But it's very early. We don't have much to go on yet. Last year, my colleagues, Giovanna Ponti, Graziano Fiorito, and others, and myself, published a sort of a commentary, a review, and we talked about sort of moving from observations of neural plasticity, that is the behavioral and neural flexibility of octopus and other cephalopods, to an argument for conscious experience, for consciousness, for the ability to have subjective experience. So, again, these animals have very impressive discriminatory and anticipatory behaviors. They can discriminate. They can make some fine discriminations, but they also can anticipate. They also seem to monitor their visual scene, which we can talk about in a bit. Cephalopods are effectively big-brained invertebrates that are confronting a temporally and spatially variable environment, okay? We have been able to identify neural substrates in octopus that may be important for subjectivity. We don't know, but it's something that we sort of have to pursue systematically. J.Z. Young, very, very early on, happened to choose the cephalopod as a model of a generic model for the nervous system. And, one may or may not criticize that approach, but it was very imaginative and perhaps the very start of the notion for the importance of looking for complex cognition in an invertebrate, and even, I dare say, consciousness in an animal like an octopus. So we have very little so far, but we have some idea of the neurophysiological dynamics and functional signatures of various behaving states in the cephalopods, but we don't have very much yet. But it's something that we should sort of bear in mind because it's an area where I think we can develop some evidence for functional signatures of conscious states in a very, very different kind of an animal. All right. So one thing that we have seen in octopuses and other cephalopods is fast, irregular brain activity. We've definitely observed compound field and evoked potentials, and that goes way, way back to Ted Bullock, among others. There are periods of widespread brain activity in cephalopods, something that you also see in the nervous systems of mammals and humans in particular during conscious processing. And now, functional analogs of cortex and thalamus. Again, I mentioned the superior frontal vertical lobe system, and I'll also add the dorsal basal lobe of the octopus and other coleoid cephalopods. But it's been suggested that these actually may be working as functional analogs of cortex and thalamus. So this is a sort of a good and fertile area sort of to begin our studies of putative awareness in a far-flung species. So let's talk about the eye. Classic case of convergent evolution. When we look at the focusing eye, the human eye on the left, the octopus eye on the right, cephalopod eye on the right, they have very similar structures. There's an eyelid in the case of the human. There's something like an eyelid, like a sphincter in the case of the octopus. They have corneas, they have pupils, they have an iris. There are ciliary muscles in the case of humans which control our ability to focus. We can see near and far because the ciliary muscles contort, they distort the shape of the lens in the eye, allowing us to focus in on things. In the octopus, it's different. The lens moves in and out more like a true camera. There's a retina. So there's a sheet of photoreceptors in the case of the human, also in the case of the octopus. There's an optic nerve and an optic ganglion. We see all of that. The difference, of course, is that in the case of the retina, a lot of processing is going on, a lot of processing of the visual signal is going on before it even hits the central brain of the mammal, of the human in this case. In the case of the octopus, there's really just a sheet of photoreceptors. There's not a sort of a layer of cells that are doing some processing after the signal leaves the photoreceptor. But the octopus's signal is going directly into the brain. Well, what's interesting is it seems as if all of the functions of the vertebrate retina, in terms of all of the pre-processing that happens, is occurring. It's been offloaded into the central brain of the octopus. So if you look at the common octopus, a sort of a cross-section of their optic lobe, you see the signals coming in from the photoreceptors of the eye, and they go in, and what's really intriguing is the nature and arrangement of these various cells in the optic lobe very much resembles the kinds of arrangements that we see in the retina of a typical mammal. And we're doing a flyby in these particular examples on the right. So you're just sort of seeing us. We've made slices of an octopus optic lobe, and we're flying through those slices and getting an idea of what the 3D sort of arrangements of the cells are. So in terms of what that gives the octopus, what that kind of eye gives the octopus, well, you can see from the spatial acuity of various animals, the octopus does rather well. It has a pretty good spatial acuity compared to the vertebrates. It lies somewhere between humans and many different birds. So there are no slouches in terms of their spatial acuity. And so we can also, let's get behind the eye and let's look at some principles of visual processing, which we find in the vertebrate, but maybe sort of more universal if we're talking about complex nervous systems dealing with the kind of information that can be gleaned from a complex visual scene if you have focusing eyes. So, first of all, in the case of vertebrates, mammals in particular, there's a retinotopic organization of the cortex, so of the striate cortex. That is to say that the spatial relations of the signals coming into different photoreceptors on that receptor sheet, that photoreceptor sheet of the eye and the retina, those spatial arrangements are preserved somehow in the brain. So cells in some areas are firing relative to a signal coming into a photoreceptor in one part of the eye versus signals coming into a photoreceptor in another part of the eye. Those spatial relations are more or less preserved. And this is very interesting because there's a suggestion that this indeed is happening in the case of the octopus and perhaps other coleoid cephalopods. There's a hierarchical representation of the world in the vertebrate visual system. We know this for a fact. It's very well studied. This also may be true in the case of cephalopods like the octopus, but we just don't know yet, although we might imagine that it's the case, and it's something we could certainly look for as our octopus neurophysiology progresses. Finally, reentrant pathways of vision. Vision is famously or infamously reentrant or recursive. That is to say that information coming from various parts of the visual cortex is communicated back and forth to different areas of the brain. There's a feed forward stream, there's a feedback stream. This is something else that we should be on the lookout for as we further characterize the anatomy, particularly the functional neuroanatomy of coleoid cephalopods like the octopus. So here's an example of the other end of things. Now, physiology and neurophysiology and neuroanatomy are key to any arguments about consciousness, I believe, in animals as far-flung as the cephalopods. But we can't forget about sort of the behavioral manifestations, or what we can watch the animal doing in relatively non-invasive sort of circumstances. So here's an instance of the common octopus in an experiment that involves so-called observational, or some people have called it social learning. I prefer observational learning. So one animal is presented with a clear plexiglass box. The box has 3 different entries into it, 3 different portals, doorways to get into the box. The box is loaded with a live crab or a shrimp. In this case, it's a shrimp, I believe. And there are seemingly three possible ways in, but only one of these 3 doors actually works. Well, the animal that's been given the box early on eventually learns through trial and error which of these ports is the working port, and will go right to that port once it has learned the nature of the box. So let's watch this really quickly. And so another animal, and this is a second animal that you're watching here. The second animal has observed the first animal solving this problem. It hasn't observed the animal learning the problem. It's observed the instance of that first animal having figured out the problem, just going right to the right entrance. So this second animal, who's watched the first animal learning, or watched the first animal getting to, it's actually, I guess it's a crab, actually will learn in a single shot. It's no trial learning. He goes to the right entrance. He's seen that the first animal got into the box by using this particular portal. And instead of trying all the others, he goes to this one. He twists it. You can sort of see his web twisting and opening up the portal. And he goes in, and that poor crab is history. Okay. So really intriguing. Well, what does this entail, this notion of observational learning in a cephalopod? Well, it entails some form of memory, of course. It entails a sophisticated visual system, probably a focusing eye to observe the details of what's going on. But it definitely indicates that the animal has the capacity for a sophisticated form of memory. Working, perhaps even episodic memory is in play. And so that's really intriguing. So this is sort of yet another strain of evidence that bears on, the issue at hand. Just to give you some background, we started developing the means of presenting these animals, of presenting octopuses with visual information, but visual information of a form that could be readily manipulated. So in contrast to presenting them with live prey species, live crabs, live shrimp, we could show them movies of live crabs or live shrimp and watch them react. And we had to first figure out whether they would approach them in the same way they approach the real thing. And once we figured that out, and by the way, they can, if look to the right and you'll see an animal appear, we've hidden the camera so the animal doesn't know that there's a camera watching. If the animal saw the camera, he'd start taking the camera apart. So he sees the crab appearing, he makes a lunge for the crab, and then if you watch really carefully, he'll stand back. He hasn't succeeded. It's quite frustrating, I'm sure, if you want to apply that word. He stands back and looks to see where the crab has gone, and then he makes another lunge at the crab. Well, that's really intriguing. And that simply suggests that we're on the right track. We can do these very tractable sorts of experiments involving visual presentations, video presentations, and we can even do photorealistic animations of virtual crabs, virtual shrimp, virtual stimuli doing various things and then watching the animal behave. I've done a number of experiments involving visual navigation. I applied the so-called Barnes maze to the case of the octopus, and the Barnes maze is quite simple, actually. It's just an arena with a bunch of different holes. All of the holes are fake except for one. That is, they're recesses, but they don't lead anywhere, except for one hole, which is the one true hole, which leads the animal into its own native water, into its own tank. Okay, so the animal has a lot of, the animal has a lot of impetus to sort of find a solution to this problem. First of all, it's exposed in the light, it's in the air, it's not in a very hospitable environment, and it has to figure out a way. So what happens in this experiment is the animal eventually learns where the hole is. We put the animal into the center of the Barnes maze in a little black box. We lift the box up. The animal starts exploring. Eventually, the animal learns the location of the true hole and goes right to the hole. And then when we shift the holes, that is to say, we move the hole to a different place, but we keep a navigational a cue in place. That is to say, the walls of this arena have different sorts of objects represented on them. In this case, it's a red plus sign. It's a red plus sign that happens to be above the escape hole. The animal learns to escape in the context of that red plus sign. When you rotate the hole but keep the plus sign in the same place, the interesting thing is the animal still goes to the plus sign. It doesn't go to the true hole. So effectively, it's learned to navigate via a landmark, which is really intriguing. Again, involves some sort of sophisticated memory faculty. All right. And then finally, here's another aspect of the story, of the memory story. Clearly, this is a case where octopuses also have to use particular kind of memory. They use working memory for this, I'm sure. There may be something like episodic-like memory as well. But we found that octopuses can learn to use a mirror to find food that isn't in their line of sight. This is work that was done with a group that I've collaborated with for 5 years now at Dartmouth College, led by Peter Tse. This work was done by Marie-Louise Keisler and her colleagues at Dartmouth. And it's a really interesting finding. It's not out yet in print, so I'm going to be very careful and deliberate about how I sort of present this. But here you see the animal first experiencing the mirror. It will eventually approach the mirror. This is a sort of an early encounter. I won't spend too much time on it. But the idea behind this experiment is simple. The animal encounters the mirror. It learns that in the mirror, it can see a crab. It can see a very potent stimulus that is not in its direct line of sight. That is to say that the crab is not directly visible to the octopus except via the mirror. So the animal effectively has to learn to use the mirror to locate the crab. In this case, it's a virtual crab, and then go after the crab. Eventually, many animals actually learn how to do this, although statistically, it's much greater than chance, but it's a little bit more variable than we would like. There's a lot of variability when it comes to octopus behavior, and that's kind of part of the game. You kind of get used to that. So here's an example. It's just a quick pilot example of the animal sort of finally learning to use the mirror. I'm going to speed this up a bit. Okay, so this is the general view from the tank. So the animal initially starts in a box, and this is just the animal encountering its world. And the box sort of isolates the animal from a direct view of the virtual prey, but it can eventually learn how to use the mirror to locate that prey. And this is really intriguing. It's work that sort of needs to see the light of day in terms of publication, but it's very, very tantalizing. I won't spend too much time on it. It's not the video isn't necessarily the best exemplar of what's going on here, but clearly some animals are using that mirror in a very effective way to find prey. So let me just go through this really quickly. I'm going to kind of give you a laundry list of what I see as breakthrough innovations in the natural history of awareness. So first of all, animals with focusing eyes, that is, eyes with lenses, eyes that have a single compartment. They're not multi-chambered. They're not multifaceted like insect eyes. They're actually true single compartment camera eyes. These animals generally tend to evince more complex and flexible behaviors than other members of their phyla, okay? Well, when you get distance vision, and I would argue that is happening in the Cambrian, in the middle of the Cambrian explosion, when you get distance vision via focusing camera eyes, you effectively buy time when it comes to far-off salient events, whether that salient event is the appearance of a predator or the appearance of a prey. All of a sudden, if you have focusing vision, you're not right up against that animal. You may not have even given that prey or predator species any kind of indication that you're there. You're far away, and you have the benefit of distance and time on your side. And so what does that mean? Well, that means you can monitor events. All of a sudden, you can monitor events. You don't have to behave reflexively. You don't have to suddenly go after the prey with a very, very poor chance of getting it, especially if it's far away. You can monitor. You can even make predictions. So it buys time. I would argue that along with this comes inhibitory circuitry. It's sort of look and monitor or plan before you leap. There are plenty of anecdotal accounts of coleoid cephalopod behavior that support this. Focusing eyes. Focusing eyes suggest brain circuitry for monitoring and prediction, and I believe new kinds of memory as well, working and episodic. Construction of detailed visual scenes, stitching together textures, contours, intensities, wavelengths, movement, other aspects of the visual world. All of that becomes important, and eventually, I would argue, binding all of that all of those submodal properties. And then you have to, if you're talking about the appearance of focusing eyes, you necessarily have to talk about the appearance of specialized brain circuitry for doing, for handling all of that visual information and, decomposing it and then integrating it into some sort of a dynamic representation. So you need to have the neural circuitry to bind submodal and even multimodal properties of the world, some sort of dynamic mapping, okay? And I just want to offer the caveat that when I put vision to the fore and I suggest that vision is very, very important in the evolution of consciousness, I'm not suggesting that animals that don't rely on vision aren't capable of conscious experience. That's absolutely ridiculous, of course. I'm suggesting, though, that vision provided an in very early on, during the Cambrian, arguably, provided an opportunity for the emergence of a very early form of primary consciousness, of sensory consciousness. And later, arguably, one could surmise that that neural circuitry was co-opted, as has happened on a number of different occasions in the case of brain evolution, to other sorts of regions and circuitry. It gets co-opted for other sorts of purposes. But in effect, the kind of circuitry that developed behind sort of the early vision, the early visual system, may have been co-opted later. So effectively, blind animals are aware. Animals that are effectively born blind are capable of conscious experience, although it might be harder to demonstrate that in some cases. But I'm just arguing that vision opened the door initially. All right. And so what follows from sensory integration and binding, working and episodic like memory, and putative reentrant or recurrent brain circuits that connect visual perception and visual memory? Well, I would say sensory consciousness. And that's where I'd like to end it. I thank you for your attention, and I would like to mention how deeply grateful we are for the great work and generous support of all of our colleagues. Thank you very much.
Thank you, David. I'm sorry, I could just three short questions, please, because we have we have so late.